and I'm going to talk to you about the Microbial Geo Program. We're structured this way. The little boxology here, I'm the acting program manager. We have a project manager that I'll talk a little bit about later on, and each program has a project manager. We have various functional groups that are associated with programs in general. In the Microbial Program, there's a group that does the draft sequencing that's run by Susan Lucas at the JGI in Walnut Creek. Once that draft is completed, we do finished sequence and assembly, led by Chris Detter at Los Alamos National Labs. For both draft and finished assemblies, Oak Ridge does automated annotation. We do QC on our finished annotation by a group run by Nikos at the JGI. Ultimately, your sequence gets submitted in a GenBank done by Sam Pitluck, and then you can view your data through the IMG portal. So, just to go back over that again, your project is authorized through a program, and I'll talk a little bit more about that in a bit. You provide us with source material, the genomic DNA. I will comment that in all the programs, oftentimes the creation of starting material is the rate limiting step. So I encourage you to ahead of time understand how you're going to isolate your material and provide it to us as soon as you can. Draft is done at the Production Genomics Facility, the JGI in Walnut Creek, and Fong, for the Microbial Program, Fong did a good job of describing what our current strategy is, which is mainly new technology based. Traditionally, and probably most of our projects that are currently in GenBank were Sanger based strictly, but over the last year in particular, we've switched to new technology platforms. Once the draft is completed, there's a quality control step that goes through and makes sure that the draft assembly has sufficient coverage and sufficient quality to move it on into a finishing process. Also at that point, the Oak Ridge folk do automated annotation on that. On the draft assembly, then it goes to a finishing team, which is physically located at Los Alamos National Laboratory in Los Alamos, New Mexico, where the draft assembly is polished, finished. By the end of that process, you have a high quality single assembly. If needed, and particularly when we change technologies, there's a quality assurance step at that point that's carried out by Hudson Alpha currently in Alabama, as Jim indicated. Once again, Oak Ridge goes back, does automated annotation. Once everything's put together, the data is submitted to GenBank, and you can view it through IMG or any other tool of your choice. So, to go back over out of this process, what are various endpoints? From the draft sequencing, we have a draft assembly and a draft annotation. That's available to the users fairly quickly, and that allows you to take a look at the data before it goes through the finishing process. Once it's finished, again, you have access to an assembly and an annotation. We do GenBank submissions on both the draft data and the finished data, and then you can move on and at that point you can use the IMG tool. I might mention that there's a workshop next door right now on the IMG tool, and I encourage you to take a look at the IMG tool through the JGI website. So how are we doing? If you take a look at completed microbial genomes released in NCBI, you can see this is worldwide. We have been the leader for the last several years. We anticipate that we will continue to be the world leader for at least the next few years. What are we doing right now? Right now we have 421 projects, microbial projects that are in the queue. We've completed about 280. So over the lifetime of the Microbial Genome Program, which as Jim indicated earlier, has been really begun in 2001 through the present, we're either completed or actively working on 700 projects. So we've done a lot of work over the last 5 or 6 years. So how do, of those 700 projects, how did they get into the process? Of most interest to you is the Community Sequence Program. I'll talk a little bit more about that in a bit. The next area is GEBA, the Genome Encyclopedia of Bacteria and Archaea. That's a JGI initiative. Those projects were identified internally and started internally. Jonathan Eisen tomorrow will give more discussion about that. And then our third and newest input is the DOE Bioenergy Centers, and traditionally we've had some DOE Genomes to Life, and this is a DOE mandate that the JGI sequence these projects. So GEBA, as I mentioned, Jonathan will be talking about this tomorrow. You can also go to our webpage and read more about it. Right now, probably a quarter of our active projects come from the GEBA initiative. So of most interest to you, how do you get projects into this program? Back to the web page, the Community Sequencing Program is the primary portal, our primary entry point for you into this system. Jim said that we have yearly calls for the CSP. For the microbial program, it's actually quarterly calls. Let me talk a little bit more about that. So, for microbial projects, right now, April 24th is the deadline for submission of projects, and then you'll be able to submit projects actually year-round, but they'll be reviewed on a quarterly basis. The review process for the microbial programs are more abbreviated than they are for the larger programs. Internal review, fairly quick turnaround. There's a scientific internal review. And we've moved to this model of doing quarterly reviews so that the users can get their projects into the pipeline more quickly. So, as I said, scientific and technical feasibility review of the proposal once that you've submitted it. When your proposal is approved, then your project is initiated by the project management office. So this is the JGI Project Management Office. We all report to Jim. I lead this group of folks. For all programs, these are the project managers, and specifically for the Microbial Genome Program, Lynn Goodwin is the project manager. And once your project is initiated, she will be your primary contact for the life cycle of the project. And in conclusion, you too can have this program. So there's the Microbial Geo Program tank, which is currently available. If you go on to, so this is published in scientific journals, but the most direct way to get to this particular, to apply for this particular job is through the LBL website. So that's it for me. Do you guys have any questions? I'd be happy to speak further about this. No questions? Sir?
Are there microbes you wouldn't sequence?
Well, is that a loaded question, Dan? Dan, our program manager. So the party line is, if it's of DOE relevance, we absolutely wouldn't sequence them. I'd say the answer is no. A prokaryotic microbe that meets the DOE mission requirements is very good for the program.
Is there any sort of a time lag between completion of the sequencing and assemblage, doing all of your magic for the primary investigator to submit it? Is there a gap in between?
So is there an embargo period? So we call that an embargo period. Let me rephrase the question. The question, correct me if I'm wrong. The question is will the investigator have an opportunity to have exclusive access to the data for a period of time before it's released to the public? Is that correct? Correct. Jim, would you care to comment?
Go ahead. Go ahead. So the short answer is yes. Our general approach is that data should be shared with the broader public as soon as possible. We encourage people to forego any embargo. Our data release policies are almost continuously in flux, balancing the needs of investigators to use their data in specific ways, and they've gone to the trouble of writing proposals and gathering DNA, which is sometimes a non-trivial effort. So for projects in which investigators have gone particular lengths to cultivate organisms that are difficult to cultivate, go to the bottom of the ocean to get materials, we believe they deserve an embargo period. So that's generally 6 months.
Does that include the...
It's project specific. You must request it, and we will encourage you in all circumstances to make data available immediately.
Sorry, does that include the data, the original sequencing data, exclusive
Yeah, so we've recently transitioned so that the read data are deposited immediately, and it's assembled data. So the embargo period starts once you've got assembled annotated data that you can actually start to analyze.
I'm sorry.
Okay. Do you sequence pathogenic organisms like anthrax?
Well, those aren't of DOE mission relevance, so the answer is that's no. Have we done pathogenic sequencing in the past? Yes.
So plant pathogens you do?
David's weekend.
Well, again, I encourage you to submit a proposal. If you can attach plant pathogens to DOE mission relevance, perhaps it's threatening feedstock, then it would be considered. Do we have mechanisms to receive genomic DNA from risk group 2 or above microbe? The answer to that is yes, and I'd be happy to talk to you about that later on. But our requirements are basically biosafety committee driven, and we're at the SL level.
Just a follow-up question on the data release policy. If we're general public, not one of the special users, and we have access to the data, is it assumed that then we can publish on that? Or should we be contacting somebody specifically?
Again, this is an area of a lot of discussion. In general, it depends a little bit on the type of project. For a prokaryotic project, most whole genomes are not now most prokaryotic whole genomes are not published as a whole genome data. People want to sequence a prokaryotic organism because they're interested in a pathway, they're interested in some aspect of the biology of that organism. A prokaryotic genome data by itself is not much more than a flying planetary program. In general, the standard across the genomics community is that the right to publish genome scale analyses of any genome is reserved to the people who are driving the project. And for larger eukaryotic programs, we still insist that that's the case. We've had a number of circumstances where data have been published, where people have done investigations on using those data, come to conclusions before the genome paper has been published. What we've usually done is put the new public investigator in touch with the PIs of the program, and one of two things happen. Either they have said, this is fine, go ahead and publish it, this is not going to steal our thunder at all, or in many circumstances, it's been an important addition to the genome paper, and a new collaboration has been formed, and they get included in the genome paper. So we encourage you to, as a courtesy, to be in touch with the owners of the data. But in general, once data are public, you're free to do whatever you want. Does that make sense? You show some sensitivity, but in general, public data, and they're there.
Yeah, basically, once it's out there.
Once it's public, it's public. Nobody can stop it from doing whatever you want.
I'm just wondering about turnaround time. So first on the quarterly review, how long for a yes, no answer? Then assuming it's yes, how long does it take to, what's your typical turnaround time for a course?
So for the quarterly submission, we hope we hope to have reviews done by the end of April, and I would guess the first week of May we'll be sending out the approvals along with the priorities. So we hope it will be not that much longer. The slowest part of any genome project is getting DNA in the door. High quality DNA that is sufficient for good library creation is the hard part. Once that's done, David, what's your
It's a product, so the estimates are probably half the projects are, once the genomic data is in the door, half the projects are probably the draft phase is 6 to 8 months. Okay, but a lot of that depends on, that would be an ideal situation. The other half of the projects tail out over maybe a year, and then once it goes to 50, that could be 2 years.